Dear all, welcome to my channel Biology Notes. Today I will discuss about the conjugation process between F plus and F minus bacterial cells. Contents of the video include general overview on genetic exchange between bacterial cells, concepts on conjugation, overview on F plus and F minus bacterial cell and F plasmid, and conjugation process between F plus and F minus bacterial cells. Genetic exchange in bacteria may happen by three different mechanisms: conjugation, transformation, and transduction. Due to exchange of genetic materials, new combinations of genes are generated. It allows bacteria to evolve and adapt to new environmental niches and to sudden changes in existing habitats. Although genetic exchanges are beneficial to bacteria, but they create serious problems for humans who are attempting to combat bacterial diseases. So, in this video, I will discuss on the conjugation mode of genetic exchange between bacterial cells. Conjugation is a process in which there is a unidirectional transfer of genetic information through direct cellular contact between a donor bacterial cell and a recipient bacterial cell. Conjugation was discovered in 1946 by Joshua Lederberg and Edward Tatum. Conjugation occurs in and between many species of bacteria, including gram-negative as well as gram-positive bacteria. For example, E. coli, Argobacterium tumefaciens, Shigella, etc. In our today's discussion, I will discuss about the conjugation process between two strains of E. coli. One strain has fertility factor or plasmid in its cell and called F plus cell, and the other strain lack the fertility factor or plasmid and designated as F minus cell. Before we proceed to the conjugation process, I will focus on the F plus and F minus cell. This is an F plus bacteria or cell. Now, why this is called F plus? Because this cell possesses an F plasmid as well as chromosomal DNA. F denotes fertility. So, F factor indicates fertility factor, or we can say F plasmid. It is a circular DNA. In the early 1950s, Esther Lederberg discovered this F factor. This F factor produces F pili or sex pili that play a major role during conjugation. This cell is F minus cell. It contains only bacterial chromosome and there is no F plasmid. Now, if we see the F plasmid, then we will see it is a circular DNA of about 100 kilobase pair. This is ORI-T region, also called origin of transfer. It is the starting point of conjugative transfer. There are two IS3 or insertion sequence 3 element and one IS2 element. Presence of one TN1000 or transposon 1000. Here, genes for replication are present. This region is known as TRA region or transfer region. This is the most important part of this plasmid. If we take a closer look, then we will see such an arrangement of different genes. There are actually so many genes, but I have shown here only those genes which are essential for the conjugation process. This is TRA gene. It helps to produce pilin proteins, which are the structural units of pili. In this region, there are three genes, Tra-K, Tra-B and Tra-P. These genes produce proteins that are components of a type 4 secretion system. This is Tra-D gene. It encodes coupling protein. This is Tra-I gene and encodes relaxase enzyme. In the next clip, I will show you how these products are essential in conjugation process between F plus and F minus bacterial cells. In this diagram, I have shown you the conjugation process between F factor possessing donor bacteria and recipient cell that lacks F factor. This is an F plus cell. It has a plasmid and a bacterial chromosome. As it has F plasmid, so it bears sex pilus. This is a sex pilus and it will play the important role for transferring the F plasmid genetic materials to the recipient bacteria. This is an F minus recipient cell. It has only bacterial chromosome. The sex pilus makes contact with F minus recipient cell. Once contact is made, the pilus retracts and brings the cells into close physical contact. The F plus cell then assemble a type 4 secretion apparatus and prepares for DNA transfer. The sex pilus is embedded in the secretion structure. The type 4 secretion system joins the two cells. A complex of proteins called relaxosome makes a cut at the origin of transfer of the F factor. As a result, one DNA strand begins to separate from other strand. The intact strand is replicated by the rolling circle mechanism. This replication is also initiated by relaxosome. Relaxase, an enzyme associated with the relaxosome, remains attached to the 5' end of the nicked strand. Other accessory proteins of the relaxosome are released. The DNA relaxase complex is recognized by the coupling factor and transferred to the secretion system. The secretion system pumps the DNA relaxase complex into the recipient cell. Finally, when the DNA enters, the F factor DNA is replicated to become double-stranded. The F minus cell or the recipient cell becomes F plus. So in this case, none of the bacterial chromosome is transferred. Only the F factor is transferred. So this is all about the conjugation mode of genetic exchange between F plus and F minus bacterial cells. If you have any query, please write in the comment section. Like and share. Subscribe the channel and press the bell icon to get notification of new video. Stay with us. Keep watching. Thank you.